Great. Okay, thanks all. And please remember to mute yourself when others are presenting. Thanks very much. All right, Doug.
Okay, I'm going to start the webinar now, and then I'm going to spotlight over to you, Anna, and it's going to be great.
Awesome. Have fun, everyone. Right. Good morning, everybody. As you're coming into this webinar space, I'd just like to say a few words on behalf of ForSpace, a few words of welcome. Bienvenue tout le monde. Welcome to this panel on climate change and natural systems, featuring the work of researchers from the Department of Biology and the Department of Geography, Planning and Environment, who are examining issues related to climate change and biodiversity. So they'll be introduced shortly. Before we go there, just tell you a few words about Concordia University's ForSpace, who are the virtual hosts for this event. We are located in downtown Tiohtià:ke, Montreal, on unceded Indigenous land. And as a physical space, or these days a virtual space, we operate as the kind of front door to the university, whereby we invite you to come in and experience any number of interactive live events. And while you're there, learn about what Concordians are working on in terms of research initiatives and course activities. So these days, checking us out on social media or our website is the best way to connect. And speaking of the digital sphere, we are currently recording this event and live streaming at CU ForSpace on Facebook. So I'll pop those links in the chat in a minute. Today, you're more than welcome to join in the conversation by sharing your thoughts and comments throughout in the chat if you feel like doing so. However, if you have a specific question you'd like our panelists to address, we do ask you to use one of the two options. Either pop it into the Q&A, making our moderators' life a little bit easier here, navigating the questions coming in, or you're more than welcome to raise your virtual hand and we can unmute you so you can speak and ask your question that way. Okay, on that note, it's my great pleasure to pass the mic over to one of our collaborators here, Jim Grant. Over to you, Jim.
Thank you, Anna, and to ForSpace for co-hosting our 4th annual Sustainability Cross Disciplines Conference, together with the Loyola College for Diversity and Sustainability and the Loyola Sustainability Research Center. So welcome to day 2 of 5. Day 1 was fantastic. This is the first session of day 2. We're gonna have 5 very intriguing talks today that I'm looking forward to. It's the talks are all about sustainability and the climate crisis is our general theme, but today will be climate change and natural systems. So we're gonna begin with 3 talks about paleo approaches. What can we learn from the past about the present? And then we'll have 2 other talks about current climate change and the effects on 2 particular vertebrate groups. So let's get started. The first 3 talks are from the St Jacques lab, and I'll introduce Dr. St Jacques momentarily, all from the Department of Geography, Planning and Environment, but also all speakers today are members of the Loyola Sustainability Research Center. So our first speaker is Alex Pace. Alex is an MSc student in the St Jacques lab and will be speaking on reproducing the conditions of past climate with tree rings in the mountains of the Gaspésie. Alex, take it away.
Good morning. Do you see my slides?
Yep.
Okay, great. So my name is Alex Pace. I am a PhD student here at Concordia, and today I will be presenting the results for my master's thesis, the subject of which was reproducing the conditions of past climate with tree rings in the mountains of the Gaspésie. So we're quite fortunate that tree rings record climate, because to have an understanding of past climate and of when the current climate is going past its natural boundaries, we need long climate records. But the fact is that in many areas, we don't have these long records, but tree rings record climate. So we could reproduce these past records past where the weather records, so from weather instruments, past where they began. Now, how does this work? Well, as most people know, trees put on a ring of growth every year, and the ring right before the bark is the current year. So we could just count back to know which year is associated to each ring. Now, the main factor that tends to control these ring sizes is actually climate. So here you could see 2 stressful years. They're slightly smaller than the rest. So this can be thought of as 2 years where they're stressed out. And specifically for my project, we're looking at moisture. So thinking about precipitation. So these are 2 years where precipitation was low and the trees grew smaller as a result. Now, by studying the many trees and in a region and averaging together, we could get something of a chronology like this, so the annual ring width over time. And by studying this pattern of thin and wide rings, we gain the understanding of past climate because we'll go and look at climate records in this region where the trees were sampled. So for example, annual rainfall. And then for the period that they overlap, we can detect a climate signal in the tree rings. So you could see the dips and the troughs are quite similar. So this is a simplification, but you get the idea. So if these periods overlap, that overlap are related, we could model that relationship and apply it to the rest of the tree rings to make a climate reconstruction. Now, the region that we did this for is the Gaspésie, which is in central eastern Quebec. It's around 9-hour drive from Montreal, and it is an extension of the Appalachians. So you can see the park that we worked with, Parc National de la Gaspésie, a provincial park, here in darker green, and surrounding it are many adjacent protected areas. So in these protected areas are some of the highest mountains of Quebec, and very interesting ecosystem. But unfortunately, the climate records here are very short. So that is why we came in to reproduce past climate using tree rings. So some pictures to give you an idea of what it's like over there. On the left, we have the view from one of the highest mountains in Quebec. And in the far background, you could actually see the St. Lawrence River. On the right, you have the Sainte Anne River, which is the biggest watershed in directly in the park. And it's the river that we actually studied with our tree rings, but I'll get to that later. And so this park is home to vast amounts of moose and actually the last population of caribou south of the St. Lawrence River. And there's only around 80 of them left. And there used to be caribou all over Gaspésie and the Atlantic provinces. So this is a very important population, and they're actually genetically slightly different from the populations on the other side of the St. Lawrence River. And finally, we also have vegetation, some flowers that exist only in the park. So all of these things make an ecosystem that's very important to protect. And one of the ways we could help protect it is by have a better understanding of past climate. Now, how did we do that? First I'll say my hypothesis. So the interior Gaspésie has experienced more hydroclimatic variability than that suggested by the short instrumental records. So hydroclimatic variability is just the part of climate that has to do with water. So how do we do it? We measure our tree rings. So to get our tree rings from live trees, we have an increment bore. You can see it, the blue thing on the left here. It is sort of like a hand drill that's hollow in the middle and allows us to get a pencil-like sample of the tree rings, which we then mount and sand and measure. Then on the right here, we could actually sample dead trees as well, which allows us to go further back in time with an electric saw. We also sand it and measure it, and we could date the rings on these ones by matching the patterns on the dead tree to the later patterns, so the older patterns in the live trees. We have a variety of sites, both in the valley and in the mountains in this park. So one of the first sites that we sampled are the Sainte Anne River cliff sites at about 220 meters of elevation. And these sites are quite interesting because a lot of our cedar trees that we sampled are growing on the rocks, sort of grabbing onto the rocks with very little soil under them. So we were trying to reconstruct moisture. So we thought these trees, since they have very little water retention under them, that we'll see the effect of climate even more in their ring growth. Another site in the valley is an old growth cedar grove where cedars were over 500 years old, which allows our reconstruction to go further back into time. And so cedars are actually the longest living species in the eastern part of the continent. And this cedar grove was excellent for our reconstruction. Then we have Mount Hogsback, which is a mountain that looks like a hog's back, and it's interesting because the top of the mountain is all this sort of rocky debris from thousands of years of freeze and thaw cycles, and black spruce trees are on top of this rocky debris, so it's unstable, they're stressed out, the water drains quickly, and you can actually see on the right here one of these black spruce trees looks quite scraggly. This should actually look, in perfect condition, it looks kind of like a Christmas tree, so you can see that it's very stressed out. Then one of our final sites is Mount Logan, which is 1,100 meters above sea level. And this is quite the intense site. The trees are all stunted as well from the conditions. And we found that it was also useful in reconstructing past hydroclimate. Now, once we sample hundreds of these trees and measure them, we get these chronologies. So we actually had 897 tree samples, and our oldest one, sorry, it was 9 weeks of sampling to get all these samples, and our oldest one was 456 years old. So these chronologies on the right, the part that's in color are the parts that we could actually use, and the parts that are in gray is when we didn't have enough samples for them to be used statistically. Now, what's important to note in these chronologies is that the peaks and troughs of each of them are sort of match one to the other. So we have multiple chronologies for some of our sites, but if you had the time to look closer, you would see that they have thin and wide rings on similar years. And this means that something is controlling them across the region, right? And the only thing that could really be is climate. And we found specifically that the part of the climate that we're related to was the moisture. And it was actually most correlated to river flow records. And this makes sense because we could use precipitation, but river flow records are actually better capture the amount of water that gets into the watershed because the rain flows eventually into the river, right? And a rain gauge tends to be less accurate. So we're using records that actually measured the amount of water in the river over time. So we had records that go all the way to 1968. And so combining these different chronologies allowed us to make our reconstruction of past hydroclimate in the Gaspésie. And this is the Sainte Anne River reconstruction. So you could see the reconstruction in black. So it goes all the way to 1805, from 2018 to 1805, and it reproduces the stream flow, so the meters cubed per second of the Sainte Anne River. In light gray shading, you can see the confidence intervals, and in red, you can see the observed data, so the actual data from the instruments that measured how much water is going through the river. And you can see that it closely matches our reconstruction, which is what we want, right? And what this shows us is that our hypothesis was actually true. So the hydroclimate in Gaspésie has been more variable than represented by the 40 or so years of the instrumental data. And this is made obvious if we look more carefully at periods of low flow. So there's only one period of low flow in the observed data, whereas we have 5 more in the reconstruction. So these are periods of 4 or more years of low flow. And then in high flow, we have only pretty much the same thing. So only 1 in the observed record and 4 in the past. And then there's also a variety of extreme lows when there was only 1 in the observed record. Now, this we're hoping is going to help the park better understand its ecosystem and understand really when the effects of climate change are actually impacting the region. So knowing when we're going past natural variability, and that this will hopefully help, since it's a river reconstruction, the salmon fisheries in the area. So the Sainte Anne River is a very important salmon fishery, and it can also help with the caribou, hopefully, given that this reconstruction is related to the amount of snow in the region. The amount of snow in the region affects how the caribou breed. Now, this was for my master's thesis. I'm doing my PhD thesis. I just started a couple months ago here at Concordia. And so the trees actually have more to tell in this region. This is my colleague Claire sampling on one of the highest mountains in Quebec, and we're finding, or we have reason to think at least, that the trees here have a signal of temperature, so that we'll be able to make a temperature reconstruction in the region and give another resource to the park to better protect this very special ecosystem. Thank you.
That's great. Thanks, Alex. And so, sorry, I called you an MSc student. I guess we haven't updated our website, PhD student.
I just started in January, so okay. I'm not insulted.
Questions from, I don't see anything Q&A, questions from panelists, or I could ask a question if, any questions to the panelists? So Alex, that's very cool. So in that reconstruction, the growth of those trees were not affected by temperature change. Is that what the really tight link in your data suggests? It's all driven by water? Is that the implication?
So for most of the response wasn't exactly the same between all the sites, but most of the sites, it was much more related to moisture. But of course, those things are related as well, right? So especially when it comes to river records, the amount of water that evaporates by the time it gets to the river is a function of temperature to some degree. So there was some temperature signal in a site or two, but not enough not something clear that would be able to make a reconstruction out of, and the moisture signal was just more significant. Whereas when we go to the different sites, like higher sites, or some of the higher sites, we actually see a clear temperature signal. And we omitted those ones from this reconstruction, and we're using them for the temperature reconstruction for my PhD.
Excellent. Thanks very much. So we'll move on, but we'll pause after the first three to talk about, what the past tells us about now and the future. So our next speaker is Dr. Jeannine St Jacques. She's a professor in geography, planning, and environment, and a core member of the LSRC. Jeannine's an expert in hydrology and paleoclimatology. I think you can see both of those aspects go on Alex's talk. Her title today is "What Can Pollen Tell Us About the Past?" Oh, there it is, Reconstructing. I'll just it's right there, nice and clear. So Jeannine, why don't you take it away?
Okay, hello everybody. I first have to say I'm not Jeannine, I'm really Claire O'Neill Sanger. This is really Claire's master's thesis. She just was unable to be here today, so I'm filling in for her. So the title of my talk is What Can Pollen Tell Us About the Past? Reconstructing Environmental Variability Over the Last 2,000 Years Using High Resolution Pollen Records from a Small Lake in Mont-Orford National Park. Before I start, I'd just like to thank our sponsors, but I'd also in particular really like to thank the organizers of the conference, Fourth Space, Loyola Sustainability Center, and particularly Rebecca Tidler. Okay, so Claire's research questions for her master's thesis were: how sensitive have the forests of Mount Orford, the sugar maple hardwood forests, been to the climate changes over the last 2,500 years, sorry. In particular, she's wondering were the forests impacted by two climate events that we know happened in the last millennium, the medieval warm period, which went from about 900 to about 1300, and it was warm, and the Little Ice Age, which went from about 1400 to 1800, and as the name says, it was cold. So she's wondering, can we pick up this in the forests? She's also wondering if she's able to pick up the effects of human activities have had on the forest for the past 500 years. So her thesis was actually just recently published in Early Online in the Holocene. So I'll be presenting these results today. So her research site is Mount Orford. Here's where it's situated in North America. And so we're in Mount Orford National Park, which is about 2 hours southwest of Montreal. And it's about 30 kilometers north of the US border. And it's right next to Magog and Sherbrooke. So her study site is actually a small lake in the park called Étang Fer-de-lance, which is right here. And Mount Orford is the mountains there, the northern part of the Appalachians. Here is the lake that she used as her study site. And the way she got at her questions is we took a sediment core from the deepest part of the lake in 2018 and carefully extruded the core from the core. And what this mud gives us is the bottom of the core is the oldest mud. The top of the core is the youngest mud. The in-between mud is intermediate in age. So the mud is basically a time sequence. We have a time sequence of mud going back in time. And it turns out that pollen preserves extremely well in mud. It's basically indestructible. So the pollen from the vegetation surrounding the lake is preserved in the mud. So by chopping up that core into sequential mud samples, we actually have sequential pollen samples of the forest as it's changed through time. And it turns out that not only does pollen preserve well, but pollen is highly ornamented, and we can take the pollen usually to species or at least to genus. And this is just examples of the types of pollen that Claire would be finding in the lake. And you can see it's all quite variable. So this allows her to reconstruct the forest composition going back in time in her mud cores. Her site, Mount Orford, this is southern Quebec, is the red stars, Mount Orford. And you can see it's located in a type of forest called the sugar maple basswood domain. It's a southern forest. It's a relatively warm type forest in Quebec. And it's located right in between two other forest types. If you go to the west, if you go down in elevation, you go to the warmest type of forest in Quebec, the sugar maple bitternut hickory domain. If you go east, you climb up into the Appalachians and you enter a cooler domain called the sugar maple yellow birch domain. So she's at a really sensitive site in between two close ecotones that are just about 25 kilometers away. And she's on a mountain. Her lake, Étang Fer-de-lance, is nestled in between two mountains in the park. So as climate change, the forest zones are going to be moving up and down the mountain too. So she dates her core using radiometric methods. So she had about 10, led to 10 dates at the top of the core, and then moving down core in this purpley blue are dates on terrestrial macrofossils. So on seeds, on leaves, on twigs, on bits of wood. And you can see that the core dates back to the beginning of the core is in 365 BC. So she's got about 2,400 years here in her 2.5 meters of her core. And for the first 1,000 years, the dates are all pretty close. That looks good. But then there's a bit of scatter in the first millennium, but it's still pretty good. So she puts, taking these dates, the dates tell her the date of a specific sample of mud. So she then fits in this red dashed line an age depth model that gives her an age for all of the samples of the mud. And she then goes on. And this is a very, very busy pollen diagram, and I'll walk you through it. This is the summary of the pollen changes, pollen percent changes she's seen in her core. So what we have on this axis is time. So we begin at 665 BC, and we go up to 2018 AD. And what the individual small graphs are showing is the percent pollen changes going through time. And what we see is there's actually definite changes going on here. No nasty straight lines. Birch is the dominant, but it doesn't really change much, so we ignore that. But where the real action is beech is the real sign of a warm forest in Quebec. And we see that during a time that we know the first millennium was warm from work done by the Americans just south of the border. We know that the first millennium and the BCs were warm. We see lots of beach, but then when we move into the cooler little ice age at about 1300, we start losing our beach. It starts declining. We see the same thing going on with the eastern hemlock. Eastern hemlock is there in the first millennium AD. It's there in the medieval warm period, which is this time period right here. But then we get into the little ice age, it starts dropping off. Yes, it's a conifer, but it's a southern conifer. So what happens to fill in these gaps in the forest is, as we start seeing at about 1000 AD, the rise of the spruce, and we also see a rise of white pine. So we definitely have something going on changing in the forests. And we see this same pattern also in something called pollen influx. So Claire is able to measure and estimate the amount of pollen going into the lake for every year. And we see in the Roman warm period, in the BCs and in the Roman warm period, high amounts of pollen influx going into the lake. We move into the Dark Ages cold period. We have a definite dip in total pollen coming in from the surrounding forests. Then when we move into the medieval warm period, we have an influx, the pollen influx goes up. And then when we go into the little ice age, it starts going down again. So we definitely see changes in the entire lake, but then it starts getting interesting. If we start homing in on the top, the recent changes, you see lots of busyness in the top, but let's home in on this. In particular, let's home in on one pollen type called that coming from ragweed. Ragweed is hay fever. This is what makes you sneeze. And it's a native plant in Eastern North America. It's a perfectly respectable native plant. It's not an invasive. However, it's well controlled in normal situations. When we start getting into European style agriculture, particularly cereal fields, grain fields, ragweed goes bonkers. And when we start seeing big spikes of ragweed, this tells us, aha, European ag. And as expected, there was no European settlement in this area until after the fall of Nouvelle-France in 1763. This was a war zone between Nouvelle-France and the English colonies, with all of the native nations on various different sides. So no European settlement until after that. And it's 1770. This is when we see this big jump in the ragweed occurring. But Claire notices, she's really sharp-eyed, and she found something really interesting. She saw that the ragweed rise, it really begins earlier. It begins at about 1600. And in fact, if we home in also on the grass, spiking in grass is also a sign of land clearance and a lot of agriculture, which makes sense. Claire also noticed that grass starts increasing, but grass starts increasing really at about 1500. So what's going on here is either we think we have dating error possible, or this is a sign of indigenous agriculture. And we know from when Champlain went through in Lake Champlain around 1612, he's commenting that the eastern shore of Lake Champlain is covered by maize fields of the Western Abenaki, the Missisquoi Abenaki. And also we know from archaeological results from the Americans that the northeastern Lake Champlain area has Saint Laurent Iroquoian settlements and maize fields. And Lake Champlain is not very far away. It's about 40 kilometers away. So what we think here is we've got a two-step process, indigenous agriculture, and then European agriculture. So we definitely see the changes that we're expecting to see from the American work from the south of the border that are saying that throughout the 2,500 years, we see a general cooling of the climate and a wetting of the climate. We see this in these overall changes of the forest dynamics, but we also see something going on with a human signature in the last 500 years. So these are the American records that are telling us what's going on in climate and also here. And just in conclusions, this is the conclusions of her thesis. Yes, forest composition changes, definite changes, increases in spruce and white pine, declines in the warm beech and hemlock, and something going on in early, possibly indigenous agriculture that we're really excited about, and that we want to go out and take another core from another area, find another student, and look at this again more closely. So that's it. Really
Thank
great
you.
masters.
Thank you, Jeannine. I think that's the clearest talk I've ever heard about mud. So thank you for that. Let's see. There was a okay, no questions in the Q&A. Any questions from the panel? Or I don't see any in the Q&A. I mean, I could ask one. Jeannine, it looked like ragweed was going down recently in the last, I don't know how accurate that, the very top.
Yeah. No, that's correct. What's happening is when Europeans first came through, they burnt and cleared down most of the forest. So what we're seeing in Quebec is as agriculture declines, we're seeing a regrowth of a secondary forest. And I should have mentioned that. And we really also see it in the maple, because we see a resurgence of the maple in the last 200 years. So Europeans come through, the forest is burnt, clear cut everywhere. And then secondary regrowth, that's the maple, and that's the decline in the ragweed. But this secondary forest is really different from the old growth forest of the past. Lots of weedy red maple in there.
Okay, very
And
cool. Any other quick questions for Jeannine now? And maybe we'll take a break after Duane and let the three of you talk to each other a bit. So thanks, Jeannine. So our next speaker is Duane Noel, who is an MSc student in the St. Jacques Lab. He's also a member of LSRC. He'll be speaking on the effects of the Pacific Decadal Oscillation on flood maxima and frequency on the North American West Coast. Take it away, Duane.
Thank you for the introduction. This project has been an extension and evolution of my honors thesis in my undergrad, and now my master's thesis, titled the project, The Effects of the Pacific Decadal Oscillation on Flood Maxima and Frequency on the North American West Coast. Flooding is a severe hydrological event caused by snowmelt and intense rain events that occur typically during the spring and summer seasons. Floods, they represent 43% of the total natural disasters in the world, as well as 47% of all weather-related natural disasters. Over 70 million people worldwide are affected by floods, and thousands of lives are lost each year due to flooding. That's why there's a need to understand why flooding tends to occur more often over time. And that's why hydrologists, they conduct these flood frequency analysis studies to study these historical flood events, as well as predict the probability of new flood events occurring. However, there's one main assumption in their analysis, is that these yearly flood events occur independent of each other, regardless of what has happened previously in the past. And it doesn't consider the effects of climate change. And as we know, with the increase of CO2 emissions in the air and intensification of our hydrological cycle, there's a potential of more frequent and greater annual floods over time. And that's why my group have analyzed the Pacific Decadal Oscillation, which is an atmospheric ocean oscillation of multi-decadal variability of sea surface temperatures in the North Pacific Ocean. And this climate oscillation, it goes through two phases. A negative cold phase, where the sea surface temperature anomalies are below zero, shown in blue, and a positive warm phase, where the sea surface temperature anomalies are above zero, shown in red. So this climate oscillation undergoes 20 to 30 years of variability between its negatives and positive phases, as shown here, where it starts in showing the winter PDO index. So it's an average from November to March. And it will start from the negative phase to its positive phase, back to the negative, back to the positive. And in the in the late 2000s, early 2000s, negative phase, and then now currently in the positive phase. So to understand whether the PDO has an influence on these annual peak floods, we first looked at if there's a correlation between our PDO index and winter precipitation, as shown on the next slide. And this slide is showing this correlation where higher precipitation during this positive warm PDO phase is shown in red, whereas this higher precipitation during the negative cold PDO phase shown in blue. And if we look through our Western North American region, we see that there are clear regional patterns where there's higher precipitation in our negative cold PDO phase in Alaska, Yukon, and British Columbia, as well as our Pacific Northwest. However, in Southern Alaska and California, there's higher precipitation in our positive warm PDO phase. So we've shown that there's a clear link between the winter PDO and winter precipitation, but we're also interested in streamflow. And that's why we have plotted our annual streamflow patterns of these extreme PDO events that occur, the extreme cold PDO events that was in 1956 against the extreme positive warm PDO events, which occurred in 2015. And this is a river in Washington where we see there are clear different changes between our seasonal maximums and our seasonal minimums shown here. So in blue is showing our negative cold event, whereas in red is showing our positive warm event. And there's clear differences between the fall maximas, the winter maximas, the spring, as well as the summer minimums. So with the patterns shown on the previous slide, as well as the current patterns that we see with our streamflow patterns, we suspect that the PDO has an influence on these annual peak floods. So our study region consists of 250 naturally flowing rivers across the Western North American margin. This is publicly available data from the Water Survey of Canada and the US Geological Survey. We have 159 active station records and 174 station records from protected watersheds. So these rivers records are protected from urbanization, regulation, diversion. And we have split up our geographic region into 3 subregions, which consists of, we've labeled the northern region, which is Alaska, shown here, Alaska, the Yukon, and British Columbia, the Pacific Northwest, which is Washington and Oregon, and California, which is right here. And to test our hypothesis, we conducted 2 statistical tests. First being permutation tests on quantile plots, QQ plots, of our annual peak floods stratified according to our winter PDO phases. So what we did with this test, we wanted to see if there's a difference in streamflow between the negative and positive PDO phases. And our second test was a 2-sided permutation t-test on the Julian days of seasonal maximum and seasonal minimum streamflow. And what we wanted to see with the t-test is to test whether the pattern of streamflow during the year changes according to the PDO phase. So our first set of results from the permutation test on our quantile plots, we see that 41% of our station records have higher annual peak floods during the winter PDO. And we've also noticed that there's a clear regional pattern that exists throughout our study region. For instance, our northern region, which we've said that it's Alaska, Yukon, and British Columbia, majority of the stations have higher annual peak floods during the negative cold PDO phase. Similarly, in the Pacific Northwest, Washington, Oregon, once again, higher annual peak floods during our negative cold PDO phase. There's a different pattern that we observed in California, where in Northern California, it behaves similarly to the Pacific Northwest, where we see higher annual peak floods here in our negative cold PDO phase, whereas in Southern California, we see that there's higher annual peak floods in the positive warm PDO phase, which is similar to what we saw in our correlation map here, where there's higher precipitation during our warm PDO phase. Now, looking at the trends in our shift of the streamflow dates across our 3 subregions, we saw that there's the PDO has and influence on our seasonal maximum and seasonal minimum. For instance, in our northern region, we see that the winter maxima and the spring maxima are the most sensitive to changes to the PDO. For instance, the winter maxima dates tend to occur much later in the season during the positive warm PDO phase, whereas the spring maxima tends to occur much later in the season during the negative PDO phase. When we compare it to the Pacific Northwest, once again, the winter maxima is sensitive to the PDO, but we've noticed something new, which is the summer minima is also sensitive to changes in the PDO. For the winter maxima, once again, it occurs much later during the positive PDO phase, whereas the summer minima tends to occur much later during the negative cold PDO phase. And lastly, when we compare it to California, we see a new pattern that the fall maxima and once again the summer minima are, those dates are most sensitive to changes in PDO. We see a later shift in the fall maxima during the positive PDO phase, whereas a later shift in the summer minima during the negative cold PDO phase. So to conclude, we've shown that the independent flood event assumption does not hold in this region. Therefore, the PDO influence our flood patterns across this Western North American margin. We've also observed that later shift dates of our winter maxima and summer minima dates occur. And we must, with this knowledge, we must consider the effects of these large climate oscillations, such as the PDO, in our flood risk assessments, as well as the planning and designing new infrastructure near our water bodies to prevent more more destructive flood events, as shown here in our urban cities. Thank you.
Thanks, Duane. That was great. Great talk. I'll remind participants, if they have a question, they can use the hand up function and we will unmute you if you'd like to ask, or you can put your question in the Q&A. Any questions for Duane? So Duane, I think we can all relate to the flood maxima. What about the minima? Why is that also important?
Well, the minima is important for areas like California where there tends to be drought conditions. So they're important for water management purposes.
Okay. And that's where most of our produce comes from, right? These are all rivers for irrigation, et cetera, agriculture. Great. Other questions for Duane? I think we can open it up. Questions on any of the first three talks? Are there any general questions anyone would like to ask?
I have a question.
Go ahead, Rebecca.
I just wanted to ask, thank you all of you for your really interesting talks. I love to learn about all of the different ways that we track climate and try to understand what's going on. And I'm just wondering if the three of you could expand on the implications of your work for future climate models and future management. Duane, you've touched on a little bit with your talk, but I was wondering about you, Alex and Jeannine, how this work can help us plan for the future or predict for the future.
Well, with respect to forest managements, what Claire's work is showing how dynamic the Southern Quebec forest is. And you sort of get the feeling that the powers that be are sort of just going to assume that we're going to have more of the present in the future. But if we're seeing this much dynamic behavior in the past, in which we know the changes are not as big as what we're going to be seeing in the very near future in this century, it seems that people are just assuming that what we have now is going to continue. But
so you mean industrially in terms of planning? I know forest management occurs over 150 years. So
the Altitude de Forêt, we're having another fight with the Legault government because the current Legault government has been sort of going, let's go for industrial forestry and damn this, damn the law on sustainable management of the forest from 2010. There seems to be this assumption that what we have is going to continue. And we're saying, hey, what we have is a fragile thing. And we're going to be hitting it a lot harder. We're hitting it a lot harder now than what the natural climate variability was in the past.
Right. Thank you, Jeannine. Alex, do you have anything to add?
Yeah, so I had a question in the Q&A about salmon fisheries and how reconstruction may help with that. And so just to review that, salmon, Atlantic salmon are endangered and they use these rivers for breeding, for laying eggs, and they're actually sensitive to lows and highs in the river, and it affects how successful their breeding is. And so those researchers studying the salmon can use this as a reference to understand past population fluctuations and know when further interventions are needed, if because climate change will eventually probably be changing the dynamics of this river. And I touched a bit on the caribou, and the caribou are actually, their breeding success is controlled in some ways by snowpack, so how much snow there is, because it prevents predators from getting to their breeding grounds, which are high up in the mountains. And so the years that there's low amounts of snow, the coyotes and wolves have a easier time getting to their young. And so this could also help understand past population fluctuations and will help us understand again when greater interventions are needed. But there's already interventions going on with the caribou. They actually have snipers that protect the calves when they're born, because they're not even born every year. Or maybe they are, I'm not sure. But so they already have snipers protecting them. But yeah, it allows anybody researching the ecosystem to sort of gain perspective. Our reconstruction rather gives them a perspective, and will inform some of the way they approach the research, or can be used as a resource for them. And it can also, reconstruction could also be used to inform models that predict climate in the future.
Thanks.
Basically, what's happening is the river reconstruction is a snowpack reconstruction. And the caribou fawns are okay if there's high snowpack, because that protects them from the coyotes and the coyotes and the bears? Alex?
Yeah.
If there's high snowpack, the coyotes and the bears can't reach the high alpine meadows where the fawns are, and the fawns are safe. When the snowpack goes down, then the fawns are vulnerable. And that's when the park actually brings out the snipers. It's a bit crazy, but they'll have some years in which they have no fawns whatsoever, and then other years in which, oh, we have 3. We must protect them at all costs. So their future does not look good, because they need that snowpack and we're losing it.
Wow. Thank you. Alex, I have a very naive question about tree rings.
Mm-hmm.
As a tree gets bigger, do you expect the rings to stay constant, or do you have to control, because they're gaining, this bigger mass as they get bigger? Do you have to control for that, or does a tree just grow allometrically anyway? So they expect the rings to be even under constant conditions. If you put a tree in a greenhouse, your optimal conditions, would those rings just stay identical as the tree aged? Is that how trees grow?
No, over time they actually get thinner. It's called senescence.
Okay.
And yeah, we account for it statistically, and it also gets averaged out as we take more and more samples. So yeah, we definitely take that into account.
Good, thank you. Brian, go ahead.
Sure. I appreciate sort of Alex and Jeannine talking about sort of the broader relevance of how this sort of paleo climate research could be of interest to like biologists and ecologists who are doing sort of on the ground work. And I was curious for Duane's work if there's kind of a similar component where are you guys trying to sort of share this work with maybe some biologists who work in the like on the west coast of the US and Canada, and like talk to them about sort of this connection between the PDO and precipitation dynamics? Because I would imagine for a lot of animals and plants even, that's possibly very important.
Well, my work in particular was our focus was more on infrastructure and buildings. So it was more for urban cities. However, the start of the PDO was due to the beginnings of the research on the PDO started with salmon production. So that's the link with biology.
Cool, thank you.
Interesting, Duane. So salmon are really important, but is now just flooding of cities the most important economic consequence of your data? Is it shifting from because of these extreme flood events?
I wouldn't say that. I think both of them have equal importance. I wouldn't say one is more important than the other, except that from a human perspective, I guess that we care more about our cities, but there's also lots of value in managing our salmon fisheries. So,
thank you, Brogan. You had your hand up.
Yeah, I actually had a question for Duane as well. Maybe you touched on this and I just missed it, but you showed that one graph with the different cycles of the cold and the warm.
Yeah.
I noticed that right now we're in a warm and the cold cycle right before was very short. Are we like technically, like, I don't know if you know this, are we supposed to be in a cold cycle right now, but because of like anthropogenic reasons, we're in a warm, or is it common to have these short cycles as well?
It's not that common to have these short cycles. Right now, it's not clear what cycle that we're in. Usually the cycles are much longer. They're about 20 to 30 years, sometimes 40, but they tend not to be short periods of 3 to 4 years, like what we see in the last decade.
Thank you.
The PDO is what we call a quasi-oscillation. It's not a perfect oscillation. Sometimes it's behaving as expected, sometimes it isn't. We do know from climate modeling work that we expect the normal climatology of the North Pacific Ocean to go all to hell around 2040 or 2050. This is what climate models are predicting. So is the sort of screwed up condition, current condition of the PDO, does it naturally go a bit screwy? It can. We see its beat going in and out in tree ring records in the region. That the PDO is a very strong signal in the tree ring records in the Pacific Northwest. And we see sometimes it's regular and sometimes it isn't. So this current rather screwed up stage, we don't know if this is a normal screwed up stage or if this is the beginning of the harbinger of that 2050 in which the North Pacific is going to be in a very strange state. Don't know. We're running at this great natural experiment. What fun.
Great, thanks, Jeannine. Well, if there are no other questions, maybe we'll move on to our last two speakers. So thank you to the first three speakers. That was wonderful. Brogan, you're up next. Brogan Stewart is a PhD student in Dr. Sarah Turner's lab in Geography, Planning and Environment, and she's also a member of the LSRC. She'll be speaking on climate change impacts on non-human primate species based on cumulative CO2 emissions. Take it away, Brogan.
Thank you. So yeah, also I just want to apologize in advance if you hear any meowing. My cat needs in this room for some reason. So just, that's what that is. She's fine though. So yeah, as mentioned, my name is Brogan Stewart, and I will be presenting on climate change impacts on potential future ranges of non-human primate species based on cumulative CO2 emissions and the resulting surface temperature increase. So greenhouse gas emissions by humans have been directly linked to global average temperature increase. The rate of this rapid temperature change likely exceeds any known period since the extinction of dinosaurs. In the past, many species have been able to shift with their preferred climate ranges. However, these shifts have usually occurred over long periods of time during which the climate was gradually changing. Here we present a new analysis of how range distributions of non-human primates, which hereafter I will just call primates, will be affected by cumulative emissions of CO2 and the resulting spatial patterns of temperature changes. First, I will go over the objectives of the study and why it is significant. Then I will describe the data we used in our model and our methods. For clarity, I will go over a specific example of a species habitat. Then I will present the global spatial results and the projected proportions of temperature change in primate habitats given the various emission scenarios. So the objective of our study was to assess the projected temperature changes over primate ranges that result from varying levels of total CO2 emissions. This is significant because it will show how spatial temperature changes in habitats are directly linked to CO2 emissions by humans. We also use pre-industrial temperatures to estimate the extent by which projected future temperature exceeds the pre-industrial maximum temperatures in each species range. So this is significant because it'll demonstrate the amount of future habitat that increases above threshold temperatures for each species and gives us a metric to predict the potential loss of future primate habitat, as well as the relative climate-induced extinction risk across primate species. So to date, most of the studies on primates and climate change are either species or region specific, whereas we performed a global scale analysis. We used the spatial data for the order primates from the IUCN Red List, which consisted of 426 species and subspecies of non-extinct primates. For the climate data, we used the Regional Transient Climate Responsive Cumulative Carbon Emissions, or the RTCRE dataset, which represents the spatial pattern of average temperature change caused by cumulative CO2 emissions. This means that the climate data is not based on a specific IPCC scenario or by year, but rather the cumulative amount of carbon emissions released into the atmosphere. The unit is degrees Celsius per teraton of carbon, where a teraton is a trillion tons of carbon. So currently we have reached a level of 0.6 teratons of carbon compared to pre-industrial levels. This has resulted in global average temperatures rising by approximately 1 degree Celsius since then. The 0.8 teraton carbon scenario will result in global average temperature increases by 1.5 degrees Celsius. The 1.0 teraton scenario, the one I will be focusing on, which corresponds roughly to 2 degrees Celsius global average temperature increase since pre-industrial levels, and represents the upper limit of the world's climate target under the United Nations Paris Agreement. For our analysis, we also projected temperature increase for 1.2, 1.4, and 2 teraton of carbon emission scenarios. So using these data, we first determined the seasonal maximum temperature experienced within each primate species range under pre-industrial climate conditions. So the pre-industrial seasonal maximum temperature was taken to represent the highest seasonal temperature that each primate species would typically have experienced prior to human-induced climate change. So for example, for the rhesus macaque, their hottest season in pre-industrial times was the summer, and still is, and the maximum summer temperature was 34.4 degrees Celsius. So we used the pre-industrial maximum temperature as their threshold temperature. And we assumed that if this maximum temperature, were to become the average annual temperature, so the average throughout the whole year for the species, then this would indicate a substantial number of days each year where temperatures are considerably higher than normal temperature ranges for this species. For comparison, the average annual temperature was 17.2 degrees Celsius in pre-industrial time. So now the current annual average is 18.4 degrees Celsius, which means that according to our model, the rhesus macaque's range is still climatically favorable for them. I will now show you an example using the Margarita Island capuchin. As you can see, their current habitat stretches from Venezuela to Bolivia. So according to our model, currently in the 0.6 teraton carbon scenario, the Margarita Island capuchin is experiencing temperatures below their threshold. So what I mean by suitable habitat on the slide is that the average temperature in their range remains below their threshold temperature, which for the Margarita Island capuchin is 28.7 degrees Celsius. So for the 0.8 teraton carbon scenario, 99.51% of the Capuchin's habitat on average remains below their threshold temperature. You can see that they are starting to experience average temperatures above 28.7 degrees Celsius in the center of their habitat. In the 1.0 teraton carbon scenario, you can see that the amount of Capuchin habitat with average temperatures above their threshold is spreading. And as cumulative CO2 emissions increase, Capuchin habitat with average temperatures below their threshold temperature decreases. So our analysis shows that many primate species could be severely impacted by continued climate warming. This figure shows the proportion of species at each location whose habitat remains below the threshold temperature in response to 1.0 teraton carbon cumulative CO2 emission scenario. So for instance, if a particular region has a primate species richness of 3, so 3 different primate species that live in that area, and remains below their threshold temperature for 2 of the primate species, then the proportion of that area becomes 0.67. Therefore, the low values on this map reflect areas where most or all primate habitat are negatively influenced by climate change, while higher values reflect areas where most or all primate habitats remain below their threshold temperatures for those species. So you can see that the majority of the lowest proportion of habitat remaining below the threshold temperature for species will occur between latitudes of 20 degrees north and 10 degrees south. This figure depicts the number of species exposed to habitat above the threshold temperature with 1.0 teraton carbon emission scenario. So as predicted by our analysis, the number of species exposed ranges from 1 to 17, depending on the location. Globally, the areas with the greatest abundance of species, which is 6 to 17 in patches above their threshold temperature, lies between 10 degrees north and 10 degrees south, as well as in Madagascar. All right, so the results of our analysis provide an approximation of the potential climate change-based compromised habitat or habitat loss for primate species. For the purpose of our analysis, we examine species exposure to climate change, and our assumptions do not account for variation in sensitivity or in adaptive capacity. So depending on the species, they may be able to respond to these pressures, or they could be more negatively affected than we predicted. Rising surface temperatures could affect the species' resting time. So during very hot times of the day, primates will rest in shade instead of doing other behaviors such as foraging. It can also affect reproduction. For instance, it has been found that baboons will have fewer reproductive cycles and conceptions during drier years and wait for more ideal temperatures to reproduce. And it indirectly affects food availability, as hotter temperatures can affect plant growth. Our results were calculated within the current primate habitat borders with a no dispersal assumption. So in the past, there was less fragmentation with primate ranges and less human infrastructure preventing primate dispersal and causing population isolation. So each of the various emission scenarios that were analyzed showed a large variation in the number of primate species experiencing differing proportions of habitat above their threshold temperatures. So notably, in the 1.0 teraton carbon scenario, which is the bar in the very center of this graph, and it represents, as I mentioned before, the 2 degrees Celsius average temperature increase for the world, we found an average of 26% of habitat area above that pre-industrial seasonal maximum temperature, so above the threshold. So a quarter of all primate species would be facing average temperatures above their threshold temperature. So our aim was to present a global summary of primate species' potential exposure to climate change. Our analysis suggests that the anthropogenic climate changes caused by cumulative CO2 emissions will have a large effect on many primate species. We found that species exposure to temperatures above the threshold temperature was likely to be highest near the equator. It would be beneficial to increase landscape connectivity in order to assist with primate dispersal. Above all, we need to reduce CO2 and other greenhouse gas emission in order to slow down the rate and to lessen the negative impacts on climate change on primates and other species as well. So climate change is not presently the leading cause of primate population decline. However, if dispersal is not an option for primates and they are no longer able to survive within their existing habitats, it may quickly become a major threat. So it is important to address and to understand the influence of climate change in order to improve our ability to implement effective short and long-term conservation measures. So here's a list of references and photo sources. Feel free to ask me for any specific ones if you're interested. I would also like to thank everyone that has worked really hard on making this a great conference. So thank you.
Thanks, Brogan. Terrific talk. Any questions for Brogan? I could ask a first one, just as others might be framing theirs or writing them. Brogan, are you assuming it's driven by the physiology of the primate? Or do you think it's underlying maybe the temperature affecting the vegetation, the fruiting, cycles of trees, or what do you think the actual mechanisms will be? Physiology of the primate themselves, or its response to other things?
That's a really good question. I think it depends on the species. So because I did like a global scale analysis, it was really difficult to just pinpoint and like say one specific thing that would be the problem. There are some especially like really small nocturnal primates that aren't very adaptive to changes whatsoever. But then there's other primates like macaques that can adapt pretty easily. Like some have even moved into cities, into India, where now generations of monkeys are being born in cities and they've completely adapted. But as I said, there are some other species that like any kind of change in vegetation, and we're just seeing a crazy decrease in all of the numbers in the population. In Madagascar, that's a big problem. As soon as little things are changing, they all of a sudden don't have a food source anymore. So, yeah.
Right. Any other quick questions now? Because we can take some at the end as well.
I think we're back.
Oh, go ahead. Jump in. I can't see the whole screen.
It's okay. I was just wondering if there's any discussion of the potential of assisted migration going into a climate future for primates.
Yeah, so I've seen some papers talking about that. And then I'm trying to remember, I went to a talk in the fall about that as well. And they're trying, but it's also really difficult because the talk I went to in the fall, they were saying that lots of them become injured because, monkeys are so, I don't know how to put this, but like crazy, and they like try to get out of cages and stuff. They injure themselves, and they're really, it's a lot really stressful on them to take a wild animal, put it in a cage, move it like a far distance, then just let it go. And it changes also because primates have such specific social structures. As soon as you remove them from where they already live and put them in a new place with potentially different individuals, that messes up the social structure, and it can really affect the like well-being of the individuals. So there is some talk about it, and they're trying it. I believe it was in South America, the talk that I had seen, but there's a lot of issues that go with it as well. So like, I think it can be a good idea, especially if we know like for islands, if like the monkeys on the islands aren't going to be able to survive on that island, then we have to somehow assist in the migration. So it's something that needs to be figured out, I think.
Thanks.
There's a quick question in the chat, Brogan, about do we know about how could you use your model to predict changes in other animal species, other mammals in particular?
Yeah, so like I looked at primates specifically because that's what I was interested in, and also we tend to use primates as like a model for humans and stuff. So I just figured, I'd look into that. But yeah, you could do it with any kind of mammal, really. You could use the same thing. So, yeah.
But no one has yet, because you're the first on primates. So that's your next chapter, I guess. There we go.
There we go.
Okay, well, maybe we should move to our next speaker, and then there'll be time at the end for other questions as well. So thanks, Brogan. It's fantastic. Our final speaker this morning is Brian Gallagher, a PhD student in Dylan Fraser's lab in the Department of Biology. Brian's also a member of the LSRC. His topic today is impacts of climate variation on salmonid population dynamics, a quantitative meta-analysis. Take it away, Brian.
Thanks, Jim. And thanks for tuning in, everyone. So I'm Brian Gallagher, and I'll be talking to you today about the topic Jim just mentioned. And before I start, I want to give a shout out to my undergraduate assistant, Sarah, who did a lot of this work that I'll be presenting to you today. So my work concerns salmonid fishes, which are a group of cold water fish species that include salmon, trout, char, and others. And as you can see from these pictures, they are just beautiful animals, and they have incredible diversity in terms of their form and function. And many of these species are famed for their spawning migrations, like this picture behind me, where they can migrate hundreds of kilometers to get back to the streams where they were born. Salmonids also support ecosystem services and large fisheries throughout their range. And harvest activities for salmonids generate billions of dollars in economic activity every year. And of course, they're also threatened by human impacts. So much so that in a recent book by the author Mark Kurlansky, he talked about how he sees salmon as a kind of metaphor, in the sense that a lot of the things that we have to do to save the earth are the same things we need to do to save salmon and their relatives. And the chief threat among these human impacts is climate change, which, as we know, is increasing temperature and redistributing precipitation all around the world. It varies quite a bit geographically, as you can tell by these maps shown here. And it's already impacting biodiversity and will continue to do so in the future. So as a result, there's been a long-standing concern that climate change will impact salmonids because, in effect, they're a cold water fish in a warming world. And as a result, there's been decades of population monitoring and published research on this topic. And the question I really want to get into is, what can all of this data tell us when we look at it all together? And in general, the ways you do this are through methods called research synthesis. And there actually is a previous synthesis that deals with the impacts of climate change on salmonids, published in 2016. And this paper is a useful starting point, but it has a lot of limitations. So, for example, it ignores many data-rich species, and it used a method of analysis called vote counting, which essentially tallies up significant and non-significant results. And this is not considered quantitative by experts. You can't really standardize and compare effects, and you can't incorporate their variance. So there's quite a, so as a result, there's still no quantitative synthesis or meta-analysis that has been done for salmonids, despite this wealth of available data. And this is one of the primary goals in my dissertation. And this work is still in progress. Today, I'll mostly be sharing with you our methods, hypotheses, and aspects of the database that we've built to get at this problem. So a meta-analysis is a study of studies, and ours started, as most do, through a systematic literature search, which we did on Web of Science. And that turned up almost 3,000 papers that we thought might document the effects of climate variables on salmonid abundance or growth. Of these, 230 were selected for full text screening based on their titles and abstracts. And of these, 109 had suitable correlations that we could extract. These were our sort of variables of interest. And so, some papers had something like this example here, where you can see that survival is negatively affected by summer temperature. After extracting this data, we ultimately ended up with over 1,000 correlation coefficients with corresponding sample sizes. And we use this to calculate our standardized effect size, which is Fisher Z, basically a transformed correlation, and its variance, which is based on sample size. After extracting this data and calculating our metrics, we subsetted our data based on whether the response variable in our correlation was related to salmonid abundance or growth, and whether the predictor was related to temperature or precipitation. So each combination of this gave us 4 data sets overall. And our goal, which is still ongoing, is to analyze each of these individual data sets with mixed effects models that weigh observations according to their variance. And this basically gives more weight to observations with larger sample sizes and is considered the gold standard for large meta-analyses. Another important aspect of meta-analysis is the incorporation of moderator variables, which explain variation in your effect size. So if we look at sort of the distribution of our effect size across our different data sets, you can see that it varies quite a bit. And we want to include these moderators to explain this, but we also want to focus on the factors that we believe are most relevant to salmonid biology. There's 3 main classes of variables that are of interest here that I'll share with you today. And these relate to taxonomy, the life cycle, and geography. So in terms of taxonomy, there's many species of salmonids with distinct evolutionary histories, geographic ranges, and thermal tolerances. And there can also be remarkable variation within species. For example, Arctic char can display 4 different morphs in some lakes, with like this example in Iceland. There's also diverse life histories and habitat use. Some species reproduce only once, others multiple times. Some migrate between rivers and the ocean, while others are landlocked in small streams that you can jump over. So the moderators in here are essentially just genus and species. And even if there's remaining variation below the species level, that might be of interest because of this diversity within species. The next set of variables relates to the life cycle. So the salmonid life cycle consists of multiple life stages, as you can see from this example with Atlantic salmon. And the transitions between these stages are generally synced with seasonal variation in climate variables. So if you look, for example, you have adults that reproduce in the fall, eggs incubate throughout the winter, and your larvae emerge in the spring, metamorphose into juveniles, which then survive the summer. So these different stages are likely to be impacted by climate change in different ways. And the moderators I want to explore for the life cycle aspect are life stage as well as age and season. And I think a large fraction of the remaining variation can be explained through geography. Salmonids occupy an enormous geographic range with a lot of different habitats within it. And the moderators that I'm interested in are latitude and altitude, which there's already evidence that they impact salmonid growth and abundance. For example, this plot here shows how with brown trout throughout Europe, populations from higher latitudes and higher altitudes tend to grow more slowly. And in addition to that, I'm interested in seasonality and historical climate. And all of this information can generally be acquired through georeferencing and querying spatial databases. However, I'm really interested in exploring an integrated measure of geography as well, that kind of encapsulates all these other factors that I've mentioned already. And the one that I've settled on to explore is the Köppen climate classification scheme, which is mapped out here. And if you overlay the geographic range of salmonids on top of it, you can see that they occupy a really remarkable range of different climates that vary in terms of their temperature, precipitation, and seasonality. And just to wrap up, I want to just go over some aspects of our database coverage. In terms of time, our estimates span over 4 decades, as shown in this plot here, but most of them come from after the year 2000. We also have a large geographic range with estimates from 16 different countries. However, I will note that the vast majority are from the US, Canada, and Western Europe. So unfortunately, our results, we already can basically tell that they won't be fully representative for the entire geographic range of salmonids. We also have data for 24 species, which is a huge improvement from previous research that's tried to get at this problem in salmonids in the past. And I'm really excited to kind of dig into a database that has this much coverage. So when this is done, this will be the most comprehensive analysis to date, synthesizing the impacts of climate change on salmonids. We have more data, more species, and a more rigorous quantitative framework than previous research. And I'm really excited to wrap up this project and hopefully share the detailed results with you next year. And I just want to leave you with the idea that the important moderator variables that we identify in this analysis can potentially help guide more effective conservation and management strategies. For example, we can figure out which species, life stages, and regions might be most vulnerable. Maybe figure out what new research needs to be prioritized. And even just figure out how predictable the impacts of climate change on salmonids are or maybe aren't, based on what we currently know. And yeah, and like I said, I'm excited to dig into this more in the future. And yeah, thanks for listening, and I'm happy to take questions.
Thanks very much, Brian. That was great. Anybody wanting to do a meta-analysis should listen to your talk. It was a great description of how to go about doing it. Questions for Brian? I could ask a quick one. You go back to 1973. Is that, one might say only 1973. Is that because of Web of Science? Could you push that back farther by looking at papers cited in your sample, if you want to see the changes?
Good question. So I think Web of Science goes back to either 1968 or 1969. So it wouldn't go back too much farther.
Right.
But I think the benefit is that some of those older papers, you know, it was easy to calculate a correlation even back then, which maybe for some more sophisticated effect sizes wouldn't be the case. So we actually got more data, I think, from like the 70s than I was expecting.
Okay.
Yeah.
Great. Other questions for Brian?
I have a quick question. Did you have 1,073 different papers, or are those is that more than one point
No,
from each paper?
It was a little over 100 papers, and we got over 1,000 correlations from those 100 papers. Like there were a couple that reported, I think, that we were able to get like 50 or more correlations from. But yeah, it varies a lot from paper to paper. I think it ranged from anywhere from like 1 to like 60.
All right, thanks. And can you begin with recommendations already that we extend research to other areas of the world? And if so, would you have, from your experience with the literature so far, recommendations about how that research should or should not be done to work better with your analysis?
Yeah, good question. So definitely. I think that there's probably more that could be done to get data from like parts of Asia, especially, and Eastern Europe. But and some of it might actually exist, but could be buried in like reports that are published in the native language, for example. So there are some limitations there. In terms of like sort of the methodology, I'm interested in seeing my data set includes papers that basically use a spatial climate gradient or temporal climate gradients, and you can actually sample along a spatial gradient much more quickly. If you have a temporal gradient, you have to like wait 5 to 10 years, possibly more, and that's not always conducive to getting funding to figuring out what's going on. So what one of the things that I'm interested in is seeing if like the results of those spatial analyses are similar to the temporal ones. And so far it appears like they are, but I can't say yet. Yes, Jeannine.
Sorry, are we open yet to general questions?
Yes, go ahead.
Okay, yeah, this is actually a question for Brogan, if she's still here. Brogan, are you still here?
Yep, I'm here.
Okay, I'm sure you went into it, but I sort of missed it. I really liked your talk. How are you judging that your habitat is becoming unsuitable for your monkeys? What is your, can you explain a little bit more about, I'm sure you gave it and it just went by with everything. But how are you judging unsuitability of habitat for your monkey?
Yeah, so it's kind of like it's an assumption, but what we did is we looked at the pre-industrial temperatures. We went to the hottest season. So let's say that was the summer for this species or whatever. And then the hottest maximum temperature experienced in the summer would be like 35 degrees Celsius. So that's the maximum, but the annual average would be something like 18. So then we said, okay, if that hottest maximum temperature becomes the average over a year, then it's climatically unsuitable for them, because if it becomes an average, that means that there's days where it's way hotter than that, most likely, or there's an extended period of time where it is that maximum temperature. So we kind of based on that, because that would be a big change in the vegetation. That'd be a change for their physiology. And like, as I mentioned, monkeys, well, all animals, like look at us, when it's really, really hot outside, we're all hiding inside in the AC. You can't work outside when it's hotter than when it's over a certain temperature. So it's kind of the same with animals. They have to rest in the shade and they can't forage or, do other behaviors that they would be doing. So that's what we based it on, is that maximum temperature becoming the average temperature.
Okay, it seems a crude metric, but a reasonable crude metric. Yeah, I'd agree. But how are you getting your pre-industrial data? Because these are your areas in the world or places where even instrumental coverage even today is very sparse. How are you getting your pre-industrial temperatures?
Yeah, so Damon would be better to ask, because I actually use his data from some of his previous students. So they did it based on the amount of CO2 in the atmosphere. They worked backwards in their models. So
okay, so
I've looked into it a bit, but I've read those papers, but it's a little beyond me, unfortunately.
So some sort of reanalysis data. Yeah, it's about all you.
They worked backwards using the model.
It sounds like sort of reanalysis data, but it's that, because I would think the instrumental data isn't really there, so I don't know what else they would do. So yeah, okay, that's sort of reasonable. Okay, thank you very much.
Mm-hmm. No problem.
Other questions, general or specific? Questions. Go ahead, Duane.
One for Brogan. My question is, did you notice that was there a difference between the different size of primates and how they're affected by these changes in temperature, and how their suitability will change over time if CO2 increases?
Sorry, different sides? Is that what you said?
Well, for instance, okay, let's say if you compare a macaque to a chimpanzee or to a gorilla, where they're affected by the changes in temperature, where they're more sensitive to the changes in temperature.
Oh, okay. Yeah. So I only looked at exposure in my study. So who would be exposed to what temperatures? I didn't go into the sensitivity or the adaptive capacity as much, just because I did do such a large analysis. So that's something that could be done in future studies, probably, is looking more closely at that. You could take this data and then try to go from there. I did read, though, some studies looking at the sensitivity of different species. They're really horrific, though, because lots of them are heating animals up to see what they can tolerate. So that's not really in my field as much. I don't like reading those papers. But yeah, as I mentioned before, so it really depends on the species. Macaques are more adaptive, whereas some of the the animals with the smaller niches tend to not be as adaptive. So it really depends.
Okay, thank you.
Sorry, I don't know if I answered that well.
It's okay, thank you.
Any other questions? Brian, I'm thinking about your species. You have a range of thermal tolerance there. Do you think some species will actually be doing better with mild warming? Some will be doing worse. And then I'm thinking about these history people here. If you push back in time, are we going to see relative shifts in species abundance?
Brian, you're muted.
Oh, wait. Oh, this was for me?
Oh, sorry. That's for you, Brian. Yes.
Oh, I'm sorry.
Think about relative abundance of these different species, from the coldest cold, the Arctic char.
Okay.
Yeah.
I was actually wondering the same question for Brogan. That's what I was going to ask.
Okay. Yeah.
Anyway, no. So I think it's going to vary a lot geographically, and I think there's evidence that in really high latitudes, that so in the far north, basically the growth and abundance of salmonids is actually limited by it being so cold. So when you have warming, it's probably going to, at least for in the short term, probably going to increase growth and abundance in many of those areas further north. However, that area is also warming considerably faster. and, is susceptible to sort of these like catastrophic events from, having whole sections of the earth slump into the river. So I think it could be beneficial in the short term, but not necessarily in the long term.
Great. Brian, did you have a question for Brogan?
Yeah, I had a similar question about Brogan's primate study, in the sense that I was wondering if there was evidence for any species in particular that maybe do well, that are maybe sort of limited by really cold weather. Like, I don't think there's too many primates in very cold habitats, but is there evidence that like there's maybe some that might benefit from warming, at least to some extent?
Yeah, so actually there is. So the primates that are the furthest north are in Japan. I don't know if you've ever seen like the snow monkeys. Those are actually the species that I study right now for my PhD. So they're the furthest north, and they are limited by the cold, but they're quite fluffy and they've adapted to it. But other than that, most primates are closer to the equator. And they, in the past, so there are papers looking at the primate fossil record that have shown that during big climate warmings, primate species, their habitats and ranges have actually expanded, and they've actually have benefited from that. But in our analysis, we actually did a no dispersal assumption, just because of how much human infrastructure is like there now, and how limited habitats of primates already are. And it's been shown that lots of different primate species won't even go close to human infrastructure. They really stay away. It depends on the species, once again. And so that's why we did a no dispersal assumption. And also in the past, the climate warming has been more gradual. So they've had time to like move with the climate change, whereas now it's happening at a rate that's kind of, I think, unprecedented. I did read a study done on South America that talks about a bunch of the different species in South America, and if they can keep up with the climate warming. So it did their dispersal rate, their potential dispersal rate, and then the climate dispersal rate that's potentially going to happen. And I don't remember the exact numbers, but it was like the climate's changing at like 8 kilometers per, like, I can't remember exactly, but then the primates was way below that. So they wouldn't be able to potentially keep up with that vegetation change. So I think it really depends on the rate that it's happening. But that's a good question. Thank you.
Cool. Thanks.
I see that Anna has appeared. So that reminds me, we're probably running out of time. So I should probably wrap this up and say thank you very much to all our speakers. Thank you for the people who attended today. What a great session. Rebecca, did you want to say something quickly before we let
Yes,
Anna?
very, very quickly, because we're back in half an hour. So that gives us time to run and grab something to eat before we talk about climate action at Concordia. So what we're doing at Concordia to address the issue of climate change. Later this afternoon, we'll also be we'll be debating what our cities should look like in the future. If you're interested in learning more about biodiversity, we have another session tomorrow morning on biodiversity and climate change, and a whole bunch of events for the rest of the week. There are 4 events every day. Anna, thank you for putting that link in the chat, which you're super efficient doing. And with that, I will let you go. Thank you to all of our panelists and to our audience, and to the Fourth Space especially for hosting. It was really great to have you all here. Over to you, Anna.
Thanks, everybody. Just a quick word of thanks, Jim, for the moderation. Brian, Duane, Jeannine/Claire, Alex, Brogan, your presentations were fantastic. Thank you so much for your delivery and engaging with the questions. It was great to hear from you. So we'll close up now. We'll see you soon. Thank you. Bye-bye.